Imagine you're looking down your stereo microscope at a perfectly expanded day 5 blastocyst. I mean, the trophectoderm is cohesive, the inner cell mass is prominent.
Right. The morphology is just absolutely flawless.
Exactly. You load the catheter, you perform this beautifully smooth transfer under ultrasound guidance, and then 2 weeks later, you get a negative beta HCG.
It's the most frustrating scenario in the lab.
It really is. And you are just left staring at the embryology report, wondering how something that looks so physically perfect could fail so completely.
Well, the answer, more often than not, is that while you were grading its structural symmetry, you couldn't hear the biological screaming match happening between that embryo and the endometrium.
Yeah, and usually when we hear the word cytokine, our reflexive association is still tied strictly to the immune system, right?
Yeah.
Like fevers, systemic inflammation, host defense.
Oh, absolutely. But if you walk into an IVF lab carrying that narrow bias, you are missing the entire conversation.
Because in reproductive medicine, cytokines are basically the invisible molecular language of life itself.
They are the underlying code. And for you, the clinical embryologist preparing for your ESHRE certification, shattering that host defense misconception is really your very first step today.
Right, because we spend our professional lives meticulously counting follicles and grading embryos.
Exactly. But every single morphological change we observe is merely the macroscopic result of a microscopic dialogue. The molecular conversations occurring right now in the follicular fluid or at the uterine epithelium are what actually dictate your clinical outcomes.
We're really moving past basic morphology into this realm of molecular competence. And that concept, molecular competence, is exactly what we need to deconstruct in our deep dive today.
It is. Because when a cycle yields excellent blastocysts, it's super tempting to just credit a flawless stimulation protocol,
Oh, totally, or like perfect culture media. But when you encounter disastrous oocyte maturation in a severe PCOS phenotype.
Or shattered embryo quality in a patient with stage 4 endometriosis.
Right, or even complete implantation failure in a hydrosalpinx case, you just cannot rely on morphological grading to explain that failure. You really have to understand the specific molecular pathologies at play.
Precisely. And this knowledge isn't just theoretical background reading for an exam. Understanding this complex cytokine milieu is the absolute foundation for the rapidly advancing field of in vitro maturation, or IVM.
Because we are no longer passive observers, right?
Yeah.
We are actively manipulating these molecular pathways on the bench.
Exactly. So to really master this material, we have to establish the foundational molecular vocabulary first, then map the intricate microenvironment of the ovary.
Follow the fertilized oocyte's transcriptomic journey right into the culture dish.
Right, and ultimately confront the molecular realities of implantation and clinical key pathologies.
Okay, so the progression from isolated protein to clinical outcome is this massive intellectual journey. Let's establish the baseline parameters immediately. When we strip away that traditional immunological bias, what actually defines a cytokine here?
So structurally, cytokines are small secreted proteins or glycoproteins. We are talking about a very specific size class, typically between 8 and 80 kilodaltons.
8 to 80, got it. And why does that specific size matter so much?
It's critical because it dictates their ability to diffuse rapidly through extracellular matrices, like the basement membrane of a follicle or the dense stroma of the endometrium.
Right, they have to physically move through the tissue.
Exactly. Their primary biological job is to mediate intercellular communication. A source cell secretes the cytokine, it navigates the microenvironment, binds to a highly specific, high-affinity surface receptor on a target cell, and triggers an intracellular signal transduction cascade.
Which ultimately alters gene transcription. But if we look at that fundamental definition, a secreted molecule that travels to a target cell, binds a receptor, alters gene expression, and that sounds exactly like a classical endocrine hormone.
It does sound similar, yes.
Right, like estradiol does that, progesterone does that. What makes the cytokine network uniquely complicated compared to standard endocrinology?
Well, it really comes down to 4 distinct functional properties that govern their behavior. The first is pleiotropy.
Pleiotropy. Okay, break that down for us.
This means a single cytokine can exert wildly different biological effects depending entirely on the specific cell type it interacts with.
Oh, wow. So the exact same molecule does different things.
Yes, take interleukin-6, for example. If IL-6 acts on a hepatocyte in the liver, it drives the production of acute phase inflammatory proteins. But if that exact same IL-6 molecule acts on a granulosa cell inside an ovarian follicle,
Let me guess, completely different result.
completely different. It functions as a critical pro-survival factor, promoting cellular proliferation and preventing apoptosis. Same molecule, completely different transcriptomic result.
That's wild. So pleiotropy essentially means the context dictates the message. One molecular tool, many different functional uses. What is the second property?
Redundancy. The reproductive axis is far too critical to the survival of a species to rely on a single point of failure,
Right. Evolution doesn't like single points of failure.
Exactly. So multiple structurally distinct cytokines can bind to different receptors but converge on the exact same intracellular pathway to produce identical biological effects. It's a backup system.
So if one specific pathway goes down, the whole thing doesn't just crash.
Right. Redundant cytokines step in to maintain the signal. And the third property is synergism, where two different cytokines bind to a cell simultaneously, and their combined cascade produces a massive amplification of the signal.
An effect far greater than just adding them together. And I assume the necessary counterweight to synergism would be antagonism, right?
You got it. Antagonism is the fourth property, where cytokines directly block, oppose, or downregulate one another. This applies the brakes, preventing uninhibited cellular proliferation.
I always conceptualize this like a massive molecular mixing desk in a sound studio. You aren't just turning up one master dial for growth.
No, you are simultaneously adjusting 50 different faders.
Exactly. And how those levels interact, the synergism pushing the bass, the antagonism cutting the treble, the redundant backups keeping the track playing, that dynamic interaction is what actually produces a viable embryo.
That analogy holds up beautifully at the molecular level, and on that mixing desk, we classify these faders into several distinct structural families. You have the interleukins, which represent the largest group.
Communicating constantly across all reproductive tissues.
Right. Then you have the tumor necrosis factors, or TNFs, which classically regulate the delicate balance of apoptosis and inflammation. You have the interferons, the colony-stimulating factors, and the chemokines.
Chemokines are crucial for driving chemotaxis, right? Essentially laying down a localized concentration gradient, like a molecular scent trail, for cells to follow during trophoblast invasion.
Exactly. And we also absolutely have to include the growth factor superfamilies in this conversation. Transforming growth factor beta, epidermal growth factor, insulin-like growth factor.
FGF as well. They are the heavy lifters of folliculogenesis. But before we get into the specific messages they broadcast, the spatial dynamics of how they broadcast are fundamentally different from classical hormones, right? They don't just dump everything into the systemic circulation.
No, they operate across 4 highly specific spatial modes. The first and most intimate is autocrine signaling. A cell secretes a cytokine into the extracellular space, and that exact same cell possesses the surface receptor to bind it.
So it immediately responds to its own signal.
Yes, it is a self-reinforcing survival loop. For example, specific subpopulations of granulosa cells secrete IL-6, and they also express the IL-6 receptor complex, creating an autocrine loop that keeps them viable under metabolic stress.
It's like the biological equivalent of writing a Post-it note to yourself so you don't forget to keep breathing.
A very high-stakes Post-it note, yes. The second mode is paracrine signaling. This is local, neighborhood communication between adjacent but distinct cell types.
So like a cell talking to a granulosa cell.
Precisely. The ECA cells secrete IGF-1, which diffuses over short distances across the basement membrane to the adjacent granulosa cells to amplify their sensitivity to FSH. It's a localized, targeted whisper that never leaves the tissue.
Then we have the endocrine mode, which is the classic systemic broadcast through the peripheral blood. But let me challenge this categorization from the perspective of an embryologist.
Okay, go for it.
If the ultimate end result is that a specific receptor gets activated on the membrane, turning on a gene, why do we actually care about the spatial origin? Autocrine, paracrine, endocrine, from a functional standpoint, isn't the final outcome identical?
We care profoundly because the spatial origin dictates exactly how we must approach clinical diagnostics.
Really interesting. How so?
If a vital survival conversation between a Thecapha cell and a granulosa cell is strictly paracrine, measuring the systemic peripheral blood serum for that specific cytokine is a completely useless diagnostic exercise.
Because the signal is consumed locally. It never makes it into the peripheral circulation in any meaningful concentration.
Exactly. If you want to know what is actually happening to that oocyte, you have to sample the immediate local neighborhood. This is precisely why follicular fluid analysis provides such a high-fidelity window into oocyte competence.
Whereas a peripheral blood draw tells you almost nothing about that specific paracrine loop. That completely reorients the diagnostic approach.
Sure.
And just to complete the spatial mapping, the fourth mode is juxtacrine signaling.
Right. Juxtacrine is unique because it requires direct physical cell-to-cell contact. The cytokine ligand does not detach and float freely.
It remains anchored to the membrane.
Yes, anchored to the secreting cell, while the receptor remains anchored to the target cell. We see this extensively with Notch delta signaling between individual blastomeres in the early cleaving embryo.
Which means you literally cannot replicate that specific developmental signal just by dropping a recombinant growth factor into your sequential culture media. The cells have to be physically touching.
Exactly. Okay, so we have established the structural families and the broadcasting modes. Let's move to the receiving end, the membrane receptors.
Because a cytokine is completely biologically inert if the target cell lacks the hardware to hear it.
And this is a highly testable area for the ESHRE exam. You must be able to map the receptor superfamilies to their respective ligands because the architecture of the receptor directly dictates which intracellular kinase cascade will be activated.
Let's start with type I receptors, the hematopoietin receptor superfamily. These are massive players.
Huge players. They include the receptors for interleukin, specifically IL-6, and for leukemia inhibitory factor, or LIF. And the absolute critical detail you must memorize here is that these type I receptors share a universal signal-transducing beta subunit.
Called GP130, right? If there is a VIP molecule for this entire deep dive, it is GP130, though.
Without a doubt. If GP130 is mutated or absent, implantation is biologically impossible, period. Moving on, you have type 2 receptors, which primarily bind interferons. You have the TNF receptor superfamily, which are homotrimeric,
forming complexes of 3 identical units and relying on intracellular TRAF adapter proteins, correct?
Yes. Then we have the receptor tyrosine kinases, or RTKs. These are fascinating because they have their own intrinsic kinase domains built directly into their intracellular tails. When EGF binds an RTK, it doesn't need to recruit a separate enzyme. The receptor itself phosphorylates its partner.
It's basically an all-in-one hardware package.
Exactly. You also have your GPCRs, which are the primary receivers for chemokines, characterized by their classic 7-transmembrane domain structure. And finally, the serine and threonine kinase receptors, which belong almost exclusively to the TGF-β superfamily.
Okay, so ligadox, the structural conformational change, happens at the receptor level. But what actually happens inside the cytoplasm? We have 4 master intracellular pathways that drive everything, don't we?
We do. Let's start with the absolute workhorse of early embryogenesis, the JAK-STAT pathway. This is initiated by IL-6 and LI binding to that GP130 subunit.
So what's the first step once they bind?
When the cytokine binds, it causes the receptor chains to cluster together. This physically brings intracellular enzymes called Janus kinases, or JAKs, into close proximity. Because they are brought so close, they transphosphorylate each other, they cross-activate.
And once the JAKs are active, they phosphorylate specific tyrosine residues on the receptor itself, creating docking sites.
Right. And this is where the STAT proteins come in to dock, signal transducers and activators of transcription. But here's where we have to be incredibly specific, because activating the wrong STAT protein is the difference between a viable blastocyst and cellular death.
STAT3 is the specific protein you want activated in the IVF lab, right?
Precisely. When STAT3 docks at the receptor, the JAKs phosphorylate it. Once phosphorylated, STAT3 undergoes a conformational change, detaches, finds another phosphorylated STAT3, and they dimerize.
And that dimer can cross the nuclear membrane, right?
Yes, it exposes a nuclear localization signal, enters the nucleus, binds to specific promoter regions, and massively upregulates pro-survival anti-apoptotic genes like BCL2. It is the ultimate cellular survival signal.
But wait, what if a different cytokine binds and recruits STAT1 instead?
That is a completely different biological outcome. STAT1 uses the exact same basic architectural pathway, JAKs cross-activating, STATs dimerizing, but STAT1 dimers bind to different consensus sequences on the DNA, turning on pro-inflammatory and pro-apoptotic genes.
So you have the exact same intracellular mechanism resulting in complete cellular destruction. It really shows how exquisitely precise this system is.
It is all about the specific docking sites and which exact STAT protein gets the phosphate group. Now let's look at pathway number 2, the RAS MAPK cascade.
Typically triggered by EGF or IGF binding to those receptor tyrosine kinases, right?
Yes. It is a brilliant cascade of sequential kinase activations. The phosphorylated RTK recruits adapter proteins like group 2 and SOS, which then activate a small G protein called RAS by exchanging GDP for GTP. RAS GTP then activates RAF kinase, which phosphorylates MEK, which finally phosphorylates ERK1 and ERK2.
It's like a biological bucket brigade just passing the phosphate down the line. But what is the ultimate phenotypic result when ERK hits the nucleus?
Cell cycle progression and intense proliferation. In the context of the oocyte, the RAS MAPK cascade is the specific biochemical pathway that slams the foot on the gas pedal for meiotic resumption. Without ERK activation, the oocyte remains indefinitely arrested.
We will definitely dissect that meiotic resumption when we discuss the LH surge. But let's move to pathway 3 first, which is frankly the dark cloud of this group when it comes to RT pathology, the NF-kappa B pathway.
The NF-kappa B pathway is the master regulator of inflammation, predominantly triggered by TNF-alpha and IL-1 beta. Under normal, healthy conditions, the NF-kappa B dimer is locked securely in the cytoplasm, held prisoner by an inhibitory protein called I-kappa B.
So it cannot enter the nucleus.
Exactly. But when a massive inflammatory cytokine like TNF-alpha binds its receptor, it activates a kinase complex that phosphorylates I-kappa B. This tags the inhibitor for immediate destruction by the proteasome.
So the guard is eliminated and the prisoner escapes.
Right. With the inhibitor destroyed, NF-kappa B is free to rush into the nucleus. And when it binds to DNA, it turns on a massive suite of pro-inflammatory machinery, cytokines, chemokines, reactive oxygen species generators.
And in pathologies like severe endometriosis and PCOS, this NF-kappa B pathway is just chronically hyperactivated, isn't it?
It is. The guard is constantly being destroyed, and the inflammatory fire alarm is stuck in the on position, creating a highly toxic microenvironment for oocyte development.
Which brings us to the necessary countermeasure, pathway 4, the PI3KK signaling pathway.
This is the master metabolic and survival pathway, heavily utilized by LF, IGF, and EGF signaling. When PI3K is activated at the membrane, it converts PIP2 to PIP3. PIP3 acts as a docking site for the kinase AKT.
And once AKT is fully active, it performs 2 critical functions simultaneously, right?
Yes. First, it activates mTOR, which drives massive protein synthesis and cellular metabolism. Second, it directly phosphorylates and thereby inhibits pro-apoptotic proteins like BAD.
It's dual action. It builds the new cellular infrastructure via mTOR while simultaneously throwing a biochemical net over the apoptosis proteins to stop them from killing the cell.
Exactly. So if you are prepping for your exam, you must have these 4 pathways: JAK-STAT3 for survival, Res-MAPK for cell cycle progression, NF-kappa B for inflammation, and PI3KKT for metabolic growth firmly locked in your memory.
Because as we apply this architectural understanding to the ovary, we see exactly how these invisible intracellular pathways physically construct a dominant follicle.
Let's examine folliculogenesis through this molecular lens. When you look at a baseline ultrasound and count a cohort of antral follicles, you are not just looking at fluid-filled sacs, you are looking at intensely active immunological and cytokine spheres.
Right, the Seika cells on the periphery, the mural granulosa cells lining the wall, the specialized granulosa cells, the resident tissue macrophages, and the oocyte itself. They are all engaging in this complex negotiation.
And we can map out exactly who is saying what. IL-1 beta is pushing general somatic cell growth. IL-6 is flooding the local environment, binding to GP130 on the granulosa cells, activating that JAK-STAT3 pathway, and explicitly commanding the granulosa cells to proliferate and strictly avoid apoptosis.
And that proliferation sets the stage for the ruthless, highly competitive process of dominant follicle selection. Every embryologist understands the concept of the FSH window. As systemic FSH levels decline during the mid-follicular phase, the developing cohort begins to starve.
Exactly. So how does one single follicle manage to survive the dropping FSH levels while the rest of the cohort undergoes rapid atresia? It all comes down to the Paracrine IGF-1 and FSH amplification loop.
This is one of the most elegant mechanisms in reproductive physiology. As systemic FSH drops, survival depends entirely on how well the follicle can process that remaining faint FSH signal.
Right. And IGF-1, secreted locally by the Seika cells, diffuses across the basement membrane and binds to the granulosa cells.
And I like to think of IGF-1 as essentially a molecular hearing aid for the FSH receptor. It synergistically amplifies the intracellular cyclic AMP response to whatever small amount of FSH is present.
That's a great way to put it. The one follicle that manages to synthesize the highest local concentration of IGF-1 effectively possesses the most powerful hearing aid. Even as the overall volume of systemic FSH plummets, this specific dominant follicle can still hear the FSH signal loud and clear,
Maintaining its aromatase activity and estradiol production, while the rest of the follicles, lacking sufficient IGF-1 amplification, just go deaf to the FSH signal and succumb to apoptosis.
That is precisely the molecular mechanism of dominance. Now let's move forward in the cycle to the critical ovulatory trigger, the LH surge. This is consistently one of the most classic high-yield ESHRE exam traps, so anyone listening needs to pay extremely close attention here.
Oh, it's the ultimate trick question. The exam will ask something like, describe the intracellular mechanism by which LH binds directly to its receptor on the oocyte membrane to trigger meiotic resumption.
And if you begin describing G proteins or cyclic AMP in the oocyte, you have fundamentally failed the question. Because the human oocyte possesses absolutely zero LH receptors, none.
The oocyte itself is completely blind to the systemic LH surge.
Completely blind. So the profound biological question is, how on earth does an oocyte arrested in prophase 1 know that the LH surge has occurred and that it is time to resume meiosis?
It requires a highly coordinated, multi-step paracrine cytokine relay. Where does the LH actually bind?
The LH surge primarily hits the mural granulosa cells lining the outer wall of the follicle because they possess abundant mature LH receptors. When LH binds, the mural granulosa cells react instantaneously by activating metalloproteinases that cleave membrane-bound precursors,
Which releases a family of soluble EGF-like factors directly into the follicular fluid, specifically amphoregulin, epiregulin, and beta-cellulin.
Yes, so the mural granulosa cells translate the endocrine LH signal into a paracrine EGF signal. That's the first baton pass in the relay.
What happens to those newly released EGF-like factors?
Those soluble EGF-like factors diffuse inward through the follicular fluid and bind directly to abundant epidermal growth factor receptors, or EGFRs, located on the surface of the cumulus cells.
The specialized cells intimately surrounding the oocyte, and this EGFR binding triggers that RAS-MAPK cascade we discussed earlier.
Exactly. The cumulus cells are now highly activated, and they execute two distinct critical physiological responses. First, they dramatically upregulate the transcription of an enzyme called hyaluronan synthase 2, or HAS2, along with proteins like PTGS2 and TNFAIP6.
And HAS2 begins pumping out a massive, highly viscous, hyaluronan-rich extracellular matrix. Morphologically, when you were looking through your microscope during an oocyte retrieval, this massive deposition of matrix is what you observe as cumulus expansion.
Right, but what is the second, arguably more critical response of the activated cumulus cells?
The second response is that they relay that RAS-MAPK activation signal directly into the oocyte itself, primarily through the breakdown of cyclic AMP and the activation of PDT3A via gap junctions.
Precisely. This sudden drop in cyclic AMP within the oocytes removes the inhibitory brake on the cell cycle. This triggers germinal vesicle breakdown, or GVBD.
The oocyte is finally freed from its prolonged dictyate arrest. It resumes meiosis, physically segregating its homologous chromosomes, and successfully transitions from prophase 1 to M1 and ultimately arrests again at M2 prior to ovulation.
That is the exact sequence.
Let me reiterate that transition explicitly for absolute clarity, because the terminology and phonetics are critical for the exam. The exact molecular consequence of this entire relay is the resumption of meiosis, moving the nuclear state of the oocyte strictly from prophase 1 to M1 and M2.
It is a stunning multicellular coordination of biology. The systemic signal starts at the extreme periphery of the follicle with LH, and through this beautiful paracrine cytokine relay utilizing amphiregulin and epiregulin, ends up completely altering the DNA architecture and meiotic state deep within the oocyte nucleus.
Transitioning it right to M1 and M2. And it does not happen in isolation. Happening right alongside this EGF relay, you have massive concentrations of LIF and growth hormone flooding the follicular fluid.
Yes, LIF is actively binding to the cumulus oocyte complex, powerfully activating both the JAK-STAT3 and PI3 survival pathways simultaneously. It acts as a molecular shield, literally protecting the structural integrity of the complex during the chaotic process of cumulus expansion.
And growth hormone is also present, drastically augmenting the local IGF-1 action, providing the massive burst of metabolic energy required for the oocyte's internal machinery to actually execute the physical separation of chromosomes during the transition to M1 and M2.
Which naturally brings us to arguably the most important pervasive family of cytokines in all of reproductive biology, the transforming growth factor beta superfamily.
If the cytokine network had a royal family regulating the entire ovarian reserve, this would be it. We are looking at over 30 different distinct ligands, all utilizing highly specific serine and threonine kinase membrane receptors.
And all relying on a unique class of intracellular signaling molecules called SMAD proteins to physically enter the nucleus and dictate gene transcription.
The sheer volume of ligands in this superfamily is daunting, but we must focus on the heavy hitters. First, you have the critical activin and inhibin endocrine axis.
Right. Activins act on the anterior pituitary to potently stimulate the synthesis and secretion of FSH. Inhibins, which are produced almost exclusively by the granulosa cells of the developing follicle, suppress FSH secretion through classic negative feedback loops.
And clinically, you monitor inhibin B as a highly sensitive secondary marker of ovarian reserve, right?
Exactly. As the functional mass of granulosa cells inevitably declines with advanced maternal age, the capacity to produce inhibin B drops concurrently, leading to the characteristic early follicular rise in baseline FSH.
But while the endocrine feedback is important, the real stars of the TGF-β superfamily for the embryologist are the local paracrine oocyte secreted factors.
Right.
For decades, classical embryology viewed the oocyte as a relatively passive, metabolically quiet passenger.
Just waiting to be nurtured by the granulosa cells and eventually ovulated. But transcriptomic data has completely inverted that paradigm. The oocyte is actually the master regulator driving the bus.
It accomplishes this by constitutively secreting growth differentiation factor 9, or GDF9, and bone morphogenetic protein 15, or BMP15.
The discovery of GDF9 and BMP15 fundamentally changed our understanding of folliculogenesis. These two factors, which often physically combine to form a highly potent heterodimer known as cumulin, are absolutely essential for follicular progression.
They act in a localized paracrine fashion, binding directly to receptors on the surrounding granulosa cells, aggressively forcing them to proliferate and differentiate.
The proof of their necessity is stark. If you genetically knock out GDF9 in an animal model, the primordial follicles activate, but they simply arrest permanently at the primary stage. The granulosa cells fail to proliferate, no antrum forms, and the follicles never reach the secondary stages.
The oocyte's own signal is required to build the house it lives in. And here is where the molecular biology translates directly into clinical pathology. Specific genetic point mutations in the human genes encoding GDF9 and BMP15 are now known to be directly associated with the etiology of premature ovarian insufficiency, or POI.
The oocyte fails to secrete the signal, the granulosa cells fail to support it, and the entire reserve undergoes accelerated atresia.
And on the fascinating flip side of that coin, certain heterozygous mutations in these exact same factors can subtly alter the ovulation quota, resulting in multiple dominant follicles instead of one.
Yes, these specific TGF-β superfamily mutations are a major established genetic driver for spontaneous dizygotic twinning in human populations. A tiny molecular alteration in a single oocyte cytokine dictates a massive population-level phenomenon like twins.
It perfectly illustrates how molecular mechanisms inextricably dictate macroscopic reality. And while we are discussing the TGF-β family's control over the ovarian reserve, we must dissect the ultimate brake pedal of the ovary, anti-Müllerian hormone, or AMH.
We measure AMH in the clinic every single day. We use it to counsel patients, predict oocyte yield, and dose gonadotropins. But what is AMH actually doing biologically within the ovarian tissue?
It is synthesized and secreted primarily by the granulosa cells of preantral and small antral follicles. Right? Its fundamental biological job within the ovary is to actively prevent the mass, uncontrolled recruitment of the primordial follicle pool.
It achieves this by significantly suppressing the sensitivity of the primordial and primary follicles to FSH. It is an active biochemical brake holding the resting reserve in check, preventing premature exhaustion.
So when you measure a high serum AMH level in a patient, you aren't just measuring a passive static marker of quantity. You are measuring the total sum of the active molecular brake being applied by that patient's entire growing antral follicle pool.
Exactly. So if we synthesize all of this, the IGF-1 loops, the EGF relay, the SMAD signaling of the TGF-β family, the complex fluid contained inside this antral follicle, the follicular fluid, is effectively a complete biochemical biography of everything that specific oocyte has experienced.
It is the ultimate liquid biopsy. When you puncture that follicle and aspirate that fluid during an oocyte retrieval, you are pulling out the exact highly specific cytokine milieu that either nurtured and matured that oocyte or pathologically poisoned it.
And analyzing the proteomic signature of that fluid gives us unparalleled predictive power regarding the oocyte's future developmental competence.
Let's correlate specific cytokine readouts in the follicular fluid with clinical embryology outcomes. Finding high concentrations of Oocyte in the follicular fluid is generally an excellent prognostic sign, isn't it?
Oh, definitely. It correlates beautifully with strong nuclear maturation, normal spindle assembly, and high rates of subsequent blastocyst development in the laboratory.
But IL-6 presents a much trickier, highly nuanced diagnostic picture, doesn't it?
IL-6 is the definition of a Goldilocks cytokine. It requires highly calibrated, moderate physiological levels. At moderate concentrations, IL-6 is highly supportive, driving that vital STAT3 pathway for granulosa cell survival.
But when the local concentration of IL-6 becomes excessively high, the signaling flips from physiological support to pathological inflammation.
Exactly. Chronically high IL-6 in the follicular fluid strongly indicates severe intrafollicular stress, and that predictably correlates with highly granular oocyte cytoplasm, poor fertilization rates, and delayed embryo kinetics.
And then, alongside the nuanced cytokines, you have the outright molecular villains, high concentrations of IL-8 and high concentrations of TNF-alpha.
If you analyze a follicular fluid sample and find highly elevated IL-8, which functions as a massive, powerful chemoattractant for immune neutrophils, or elevated TNF-alpha, you are almost certainly looking at the biochemical signature of a patient with severe endometriosis or highly inflamed PCOS.
And the clinical correlate for the embryologist is immediate and severe. Highly dysmorphic oocytes, thickened zona pellucidae, drastically low maturation rates, and completely shattered developmental competence.
We will dig deeply into the specific destructive mechanisms of those pathologies shortly, but the foundational takeaway here is that the microenvironment definitively dictates the oocyte's absolute potential before sperm ever enters the picture.
That perfectly sets the stage for our transition. We've carefully aspirated the follicle, we have isolated our cumulus oocyte complex, we denude it, we fertilize it via ICSI or standard insemination, and we place it into our highly controlled incubators. We are moving our focus from the ovary to the in vitro culture dish and the earliest stages of embryogenesis.
And the critical paradigm shift that every embryologist must make here is realizing that the developing embryo is absolutely not just sitting passively in your microdrop, silently soaking up pyruvate and amino acids.
From the very first cleavage divisions, long before genome activation is fully complete, the human embryo is an aggressive, active cytokine-secreting entity. It's constantly attempting to modify its own microenvironment.
It begins so incredibly early in development. By the 2-cell stage, the cleaving embryo is already actively synthesizing and secreting platelet activating factor, or PAF.
And PAF acts as a highly potent autocrine growth factor. The embryo secretes it, binds it, and uses the signal to tell its own blastomeres to survive, simultaneously ramping up critical mitochondrial metabolic activity.
And remarkably, at that exact same developmental moment, the 2-cell stage, the embryo begins transcribing and expressing surface membrane receptors for LF.
Which means the human embryo is biologically hardwired and ready to receive exogenous cytokine help almost immediately after fertilization. By the 8-cell stage, as it approaches compaction, it is heavily expressing epidermal growth factor receptors.
This timing is exactly why modern, sequential, and continuous culture media formulations are designed the way they are. When you read a media composition sheet and see recombinant EGF or G-CSF included, it's not there by historical accident.
It is engineered specifically to bind those newly expressed receptors, activate the RAS, MAPK, and PI3K-KT pathways, suppress early blastomere apoptosis, and mathematically push the final blastocyst cell counts higher.
Since we are focused on the IVF lab, let's execute a deep dive into the 2 absolute VIP cytokines of pre-implantation embryo culture, LIF and IL-6. We already established that LI is technically part of the larger IL-6 structural superfamily.
We know they both mandate the use of that crucial GP130 receptor subunit to transmit their signal. Walk us through the precise cascading mechanism of exactly what ELV is doing to that embryo to build a blastocyst.
When a molecule of LIF, whether secreted autocrinely by the embryo itself or supplemented exogenously in the culture media, binds to the highly specific LIF receptor alpha chain and the shared GP130 subunit on the embryo's surface, it triggers an immediate conformational change,
Which activates the intracellular JAK enzymes.
Right, and the JAK specifically target the STAT3 protein, and this is where the biochemistry demands precision. The JAK's phosphorylated phosphorylates STAT3 at one very specific highly conserved amino acid residue, tyrosine 705.
Tyrosine 705. The phosphorylation of tyrosine 705 is the biochemical trigger. It allows STAT3 to dimerize, expose its nuclear localization sequence, and flood into the nucleus.
Yeah.
And what is the specific phenotypic result of that in a human blastocyst?
It provides massive targeted survival support specifically for the inner cell mass, completely suppressing apoptosis in these highly sensitive pluripotent cells while simultaneously promoting the robust differentiation and proliferation of the trophectoderm.
LIF is, quite literally, the molecular architect that constructs the structural integrity of the blastocyst.
And the definitive proof of how universally vital this pathway is comes from elegant knockout models. A female mouse that has been genetically engineered to lack the LIF gene is completely 100% infertile.
She ovulates perfectly normal oocytes, they fertilize normally in vivo, and they even cleave. Right.
Yes, but they absolutely cannot implant. The process completely arrests. LIF is not just supportive, it is an absolute biological necessity for reproduction.
Now, IL-6 operates alongside LIF, and it obviously shares that vital GP130 signaling subunit. But it has a fascinating evolutionary trick up its sleeve, doesn't it? It utilizes a unique mechanism known as trans signaling.
Yes, trans signaling.
I've always thought of trans signaling as a molecular master key that bypasses normal security protocols. Can you explain the structural mechanics of how this actually works?
Trans signaling is a brilliant biological workaround. Under normal classical signaling rules, for a cytokine like IL-6 to affect a target cell, that specific cell must possess a membrane-bound IL-6 alpha receptor to catch the ligand before passing it to GP130.
Makes sense.
However, the body naturally produces a soluble, free-floating version of this IL-6 receptor, often cleaved from membranes or alternatively spliced, which circulates freely in the blood and bodily fluids.
Okay, so the receptor is just floating around unattached.
Exactly. IL-6 can bind tightly to this free-floating, soluble receptor in the fluid. This newly formed combined IL-6 soluble receptor complex can then float over and directly dock with and activate GP130 on the surface of target cells that do not even possess their own membrane-bound IL-6 receptors.
It is exactly like IL-6 finding a security guard in the parking lot, grabbing his high-level access badge, and using it to walk straight to restricted buildings it normally has absolutely no access to.
That is an excellent way to conceptualize it. It bypasses the requirement for localized receptor expression. It dramatically expands the anatomical reach and physiological influence of what IL-6 can achieve.
Particularly during the highly complex crosstalk of implantation. Because the embryo does not simply drift down the fallopian tube and wait passively until it physically bumps into the uterine wall to introduce itself, right?
No, it engages in highly orchestrated advanced communication. It starts aggressively knocking on the molecular door of the endometrium days before it ever arrives in the cavity.
And the most famous of those advanced signals is, of course, human chorionic gonadotropin, HCG. The entire world knows HCG as the basis of the over-the-counter pregnancy test, but its actual biological purpose at the moment of blastocyst expansion is profound. It serves as a systemic rescue flare.
It is the ultimate survival signal. The expanding blastocyst secretes rising concentrations of HCG specifically to rescue the maternal corpus luteum from its programmed demise.
Because if the corpus luteum undergoes luteolysis and dies, systemic progesterone plummets, the endometrium destabilizes and sheds, and the embryo is catastrophically washed away.
Right. HCG acts on the LHCG receptors of the corpus luteum to prevent that. But furthermore, HCG also acts locally, directly on the epithelial cells of the endometrium to powerfully promote receptivity and vascular permeability before the embryo even touches down.
And it's not relying on HCG alone. The embryo is simultaneously pumping out colony-stimulating factor 1 or CSF1 to actively recruit highly specialized uterine macrophages to the implantation site and to promote the invasive capacity of its own developing trophoblast cells.
Furthermore, it secretes interleukin-1 alpha, which acts in a localized paracrine manner to specifically upregulate a crucial adhesion molecule on the surface of the maternal endometrium called integrin alpha V beta 3.
The embryo is literally modifying the structural surface of the uterus, telling it, put down the biological Velcro right here because I'm coming in.
It is a brilliantly orchestrated, multi-front transcriptomic campaign designed to ensure the landing zone is structurally and biochemically ready, which perfectly bridges our discussion into the most complex and poorly understood phase of human reproduction, implantation and its associated clinical pathologies.
This is the critical bottleneck. This is where the entire cycle, all the stimulation, the precise embryology, the genetic testing, all comes together or completely falls apart. We are talking about the window of implantation.
In a highly idealized standard 28-day cycle, we define this as days 20 through 24. For decades, morphologists looked exclusively for the physical appearance of pinnipodes, these dome-like epithelial projections on the uterine surface.
But we now know that morphological appearance is insufficient. The window opening is driven by a highly specific transient cytokine signature.
And the absolute undisputed king of that transcriptomic signature is, once again, LIF. The expression of LIF in the endometrial epithelium surges and peaks exactly on day 20 of a receptive cycle.
If that specific LIF peak is blunted, delayed, or absent, the window of implantation physically does not open. The pinnopods may be present, but the biochemistry is locked, period.
However, LF does not act alone. It is supported by a massive localized cast. Interleukin-11 is heavily upregulated to drive the massive physical decidualization of the underlying stroma. Interleukin-15 is secreted specifically to regulate and pacify the dense population of uterine natural killer cells,
Actively instructing them not to mount a cytotoxic attack against this highly invasive, genetically semi-allogeneic embryo. This precise, orchestrated transcriptomic window is exactly what modern clinical diagnostic tools like the ERA, the endometrial receptivity analysis, are attempting to map, correct?
Exactly. The ERA relies on quantifying that precise molecular signature. We are taking a biopsy and looking at the specific gene expression of these cytokines to determine if the biological window is displaced relative to the chronological days of progesterone administration.
But let's look at the mechanics of what happens when the window is successfully open and the embryo actually begins to invade. The maternal side must undergo decidualization.
This is not just a mild change. It is the massive physical morphological transformation of the endometrial stromal fibroblasts into secretory epithelioid decidual cells.
And this transformation is initially driven by the rise in systemic progesterone and local cyclic AMP, which together activate massive transcription factors, specifically FXO1 and HAND-D2.
And working directly within that framework, IL-11 is actively phosphorylating STAT3 to physically alter the cytoskeletal shape and secretory function of the stromal cells.
Clinically, we confirm that this massive transformation has occurred by measuring specific decidual secretory markers like prolactin and insulin-like growth factor-binding protein 1 or IGF-BP1.
The decidualization prepares the ground, but the invasion of the trophoblast cells themselves has to be governed by incredibly strict parameters. It is an evolutionary Goldilocks zone.
It is a razor-thin margin of safety. You inherently need deep, aggressive invasion by the extravillous trophoblasts to successfully remodel the maternal spiral arteries and establish a highly robust, low-resistance blood supply for the fetus.
But you absolutely cannot have the trophoblast burrowing uncontrollably straight through the endometrium and deep into the myometrium like a metastatic carcinoma, which leads to life-threatening conditions like placenta accreta.
So the microenvironment establishes a dynamic equilibrium. On one side, you have potent pro-invasion cytokines, high levels of EGF, IL-6, and carefully calibrated low levels of PGF-β aggressively pushing the trophoblast forward, digesting the matrix.
And on the exact opposite side, you have the localized anti-invasion signals pushing back, creating a biological wall. As the trophoblast invades deeper, it encounters rising gradients of highly concentrated TGF-β, massive localized TNF-alpha, and a highly specific inhibitory protein called DKK1.
These act as the definitive molecular brakes. They bind to the leading edge of the invading trophoblasts and forcefully command them to cease invasion. Stop here. Do not penetrate the myometrium.
But in the darkness of the uterine cavity, how do the invading trophoblasts even know which precise anatomical direction to grow? They have to find and remodel the spiral arteries.
It is driven by chemotaxis, isn't it? The trophoblasts are literally sniffing out a localized chemical scent trail.
Yes, the mechanism is beautiful. The specialized cells surrounding the maternal spiral arteries actively lay down a highly concentrated chemical gradient of a specific chemokine called CXCL12.
And the advancing extravillous trophoblasts highly express the specific membrane receptor for that chemokine, which is CXCR4.
The trophoblasts literally follow the increasing concentration gradient of CXCL12 straight toward the blood vessels, and as they execute this aggressive invasion, they must continuously pacify the maternal immune system.
They do this by heavily expressing a unique, non-classical major histocompatibility complex molecule called HLA-G.
Correct. HLA-G interacts directly with the highly specific KIR receptors killer immunoglobulin-like receptors located on the surface of the aggressive uterine natural killer cells.
When HLA-G binds KIR, it essentially whispers a profound biological password that says, "Do not trigger cytotoxicity, I belong here." It creates a localized zone of profound immune tolerance.
It is a breathtakingly complex, high-stakes molecular negotiation occurring over a matter of millimeters. But what happens to our IVF outcomes when that localized negotiation table is already on fire before the embryo ever arrives?
We have to address the clinical pathologies that embryologists confront daily, specifically polycystic ovary syndrome and severe endometriosis.
From a molecular standpoint, both of those highly prevalent conditions are defined fundamentally by catastrophic cytokine dysregulation. Let's look at the PCOS phenotype. These patients suffer from a state of low-grade, highly systemic chronic inflammation.
Remember that NF-kappa B inflammatory pathway we mapped out earlier? In the granulosa cells of a severely afflicted PCOS patient, that specific pathway is chronically hyperactivated.
The IKB inhibitor is constantly degraded, and the inflammatory machinery is stuck permanently in the on position. And the localized transcriptomic readout in the follicular fluid during their retrievals is incredibly stark.
We routinely see massively elevated levels of interleukin-12, which directly negatively correlates with the recovery of competent mature oocytes. But I want to push back on the fundamental pathology here, or at least ask the question that every single embryologist wonders when they're trying to denude and inject these dark, highly granular, fragile oocytes. Are these heavily elevated cytokine, the IL-12, the massive spikes in TNF-alpha, are they just an immunological byproduct of the disease, like smoke billowing from a systemic fire?
So that's a common thought, but no.
Or are these cytokines the actual physical fire doing the physical burning?
They are the fire. They are the highly active, destructive agents of damage. In severe endometriosis, for example, the local microenvironment and the follicular fluid are completely flooded with pathological levels of IL-8 and TNF-alpha.
IL-8 is a highly potent chemoattractant. Its primary job is to aggressively drag massive numbers of neutrophils into the tissue, driving the pathological, unregulated blood vessel growth, the angiogenesis that sustains the ectopic endometriotic lesions.
Exactly. But the elevated TNF-alpha, it is fiercely, directly cytotoxic. If you take perfectly healthy, donor-quality oocytes and expose them in a controlled culture dish to the exact micromolar levels of TNF-alpha routinely found in an advanced endometriosis patient's follicular fluid, the TNF-alpha directly attacks the oocyte's internal machinery.
It massively induces the rapid accumulation of reactive oxygen species, completely destabilizes the delicate meiotic spindle apparatus, and induces catastrophic mitochondrial dysfunction.
It physically destroys the oocyte's developmental competence from the inside out. So measuring these cytokines isn't just looking for a marker of poor quality. You are literally identifying the cellular executioner.
And the pathology is not confined to the ovary, which is why treating these patients is so difficult. In both PCOS and endometriosis, this wave of systemic chronic inflammation travels directly down to the uterine cavity and significantly suppresses the crucial endometrial expression of RAVA during the luteal phase.
So even if the embryologist manages, through meticulous culture and ICSI, to generate a euploid, morphologically top-quality day 5 hatching blastocyst from one of these patients, the clinician is blindly transferring it into a highly inflamed uterus where the primary key required to unlock the implantation window, the Early-Wave peak, is missing or pathologically suppressed.
Which provides the exact molecular explanation for why we see significantly lower sustained implantation rates and higher biochemical loss rates, even when transferring PGTA-tested euploid embryos in these specific patient populations. The seed is genetically perfect, but the soil is molecularly toxic.
Exactly. So, given this vast landscape of pathology, what do we actually do about it in the lab? How are we translating this dense bench science into actionable art biomarkers and targeted therapeutics?
On the diagnostic biomarker front, we already rely heavily on AMH and Inhibin B to accurately judge the resting ovarian reserve and predict our response to stimulation. But the true, rapidly approaching frontier in embryology is non-invasive embryo selection.
For decades, we have discarded the spent culture media, the tiny 25 microliter drop of complex fluid the embryo lived in for 5 days. But we now realize that drop is a goldmine.
Right, because if the embryo is highly active, secreting autocrine and paracrine factors as we discussed, it is constantly leaving a highly specific, measurable proteomic fingerprint in that media drop.
Precisely. Advanced laboratories are developing high-sensitivity assays to accurately measure the minute concentrations of LIF, IL-6, soluble HLA-G, and early HCG that the individual embryo successfully secreted into that microdrop over 120 hours.
Quantifying that precise secretory profile correlates incredibly strongly with the true underlying physiological quality of the blastocyst and its actual implantation potential in vivo.
It offers the embryologist a highly objective molecular layer of selection that goes far beyond simply looking at the static morphological density of the trophectoderm or the expansion of the blastocoel cavity.
It is the ultimate shift from observing form to observing actual molecular function. What about active therapeutics? This area is heavily tested on the ESHRE exam, particularly regarding the precise, active manipulation of in vitro maturation culture systems.
Yes. For in vitro maturation, we are no longer just dropping immature germinal vesicle oocytes into standard media and crossing our fingers. We are actively biochemically supplementing the IVM media to artificially recreate the highly complex Paracrine LH relay and the vital survival signals of the intact follicle.
Adding highly purified recombinant LF to the IVM media at precise concentrations of 10 to 100 nanograms per milliliter directly rescues highly fragile, immature oocytes.
The exogenous LF binds the GP130 receptors, aggressively activates that STAT3 pathway we mapped out earlier, and stabilizes the oocyte's internal machinery, preventing premature atresia during the extended culture period.
And we aggressively supplement the media with EGF as well to drive the meiotic resumption, correct?
Yes, recombinant EGF is typically added at concentrations of 10 to 20 nanograms per milliliter. This exogenous EGF mimics the natural amphiregulin and epiregulin paracrine signal.
It binds directly to the abundant EGFRs on the attached cumulus cells, forcibly driving the breakdown of the germinal vesicle and forcefully pushing the oocyte out of prophase 1 through M1 and successfully arresting it at M2.
This targeted molecular supplementation is absolutely vital for success in complex fertility preservation cases where we are forced to retrieve highly immature oocytes under extreme oncological time pressure.
Or in severe PCOS cases, where you might retrieve 30 follicles, but 60% of them are arrested at the germinal vesicle stage. The translation is incredible, and clinicians are also actively tackling the endometrial side of this equation, attempting to artificially force the window of implantation open.
They are. For highly complex patients suffering from chronically thin linings or recurrent, unexplained implantation failure despite haploid transfers, progressive clinicians are utilizing targeted intrauterine infusions of G-CSF granulocyte colony-stimulating factor.
Administering G-CSF directly into the uterine cavity strongly stimulates localized endometrial regeneration, heavily recruits supportive macrophages, and actively forces the necessary localized expression of LIF.
Furthermore, we are seeing the widespread clinical adoption of PRGF, or platelet-rich growth factors. By centrifuging the patient's own blood and isolating the platelets, clinicians can directly bathe the highly resistant endometrium in massive concentrations of autologous EGF, IGF, and vital pro-survival cytokines to aggressively prep the biochemical landing zone prior to transfer.
It's deeply satisfying how direct and actionable the clinical translation is. We map the missing intracellular pathway, we synthesize the deficient cytokine, we add it directly to the culture dish or infuse it into the uterus, and we attempt to rescue the cycle from molecular failure.
We have covered a truly massive landscape of molecular biology today. Let's briefly consolidate the absolute core takeaways before we wrap up.
I'll start. If you take away nothing else, you must remember that the cytokine network is highly nonlinear. You cannot manipulate one single signaling molecule like IL-6 without triggering massive cascading effects across redundant and antagonistic pathways.
Absolutely. You must also remember the fundamental absolute dominance of the GP130 receptor subunit and its downstream JAK-STAT3 intracellular pathway in actively keeping both developing oocytes and pre-implantation embryos alive under extreme metabolic stress.
And do not forget the intricate elegance of the LH paracrine relay. The ESHRE exam will test this. The oocyte has absolutely no LH receptors. It relies entirely on the mural granulosa cells to translate the endocrine LH surge into localized soluble EGF-like factors.
Those factors bind the cumulus cells, triggering the exact signaling cascade that forcefully pushes the oocyte from its dictator rest in prophase 1 through M1 and finally to M2.
And finally, remember the absolute non-negotiable biological necessity of a LIF for establishing pregnancy. Without the highly localized day 20 peak of LF expression, The window of implantation remains permanently boarded up.
The highly active blastocyst can knock all at once with HCG, PAF, and IL-1 alpha, but without ILF and the resulting STAT3 activation, the biological door to the endometrium will simply not open. But as we conclude, I want to leave you with one final, slightly provocative thought to carry into your next intense study session or your next shift in the lab. Let's hear it. We have just spent an hour mapping out extraordinarily complex intracellular signals. Cytokines are the master instructions. They are the architectural blueprints, the inflammatory fire alarms, the biochemical green lights. But to actually execute these commands, to physically manufacture the massive hyaluronan matrix during cumulus expansion, to physically organize the tubulin and segregate the massive chromosomes during the transition from M1 to M2, to rapidly, continuously cleave a single massive cell into a highly organized 100-cell blastocyst that requires a staggering continuous supply of physical energy.
That is a critical perspective. A perfectly transmitted molecular signal commanding a cell to divide is completely useless if the cell fundamentally lacks the basic structural bricks, the amino acid mortar, and the raw electricity to actually run the machinery.
Exactly. The most elegant, perfectly timed cytokine signals are utterly meaningless without the deep, highly robust cellular metabolic machinery to physically power them. Where does the ATP actually come from to fuel that STAT3 translocation?
How does the early embryo shift its metabolic preference from pyruvate to glucose as it compacts? That highly complex metabolic engine operating under the hood is what actually determines if the cytokine signal translates into life.
So as you go back to the lab, looking down through the heated stage at that culture dish, remember that you are not just looking at static morphology or simple grading algorithm. You are looking at a raging, invisible molecular conversation, a vast symphony of cytokines telling those cells exactly what to do.
The ultimate question is, does the embryo possess the metabolic energy required to answer the call?
That is the perfect transition point, and a vital concept for every embryologist to master next. Thank you for joining us on this exhaustive journey. Take this perspective, this deep molecular lens, into your next clinical shift. Apply it to your ESHRE preparations, and until next time, keep looking deeper.
Thank you sincerely for your patient listening and dedication to mastering clinical embryology. It's professionals like you who advance our field every day. If this content added value to your practice, a simple like helps it reach more embryologists and assisted reproductive technology specialists worldwide. Please subscribe and turn on notifications so you never miss updates on lab protocols, cryopreservation tips, and guideline-driven insights. Your support fuels more free, high-quality resources for our entire assisted reproduction community. Let's grow this channel together for better patient outcomes.